Hello everyone and welcome back to the food microbiology series of videos. This is going to be the second part of the food hygiene and sanity videos we started last time. In this video we're going to view the other types of pathogenic bacteria as well as viruses and prions, and a little bit of the monitoring and the identification of the microorganisms by the industry. So let's begin. We have the other foodborne pathogenic bacteria, which are the Campylobacter, gram-negative, highly motile, very proaerophilic. Indeed, it's a microaerophile. The jejuni species, the Campylobacter jejuni, comes from the bird, and there is a high frequency of that. And the Campylobacter coli comes from the pigs, and it's a very low frequency in that. We're going to focus on the jejuni. And the Campylobacter jejuni is a catalase and oxidase positive, mesophilic. It likes hot temperatures. It can cross membranes. Is sensitive to salinity, freezing, low pH, and it can create biofilms. It has many virulence factors such as the flagella, the capsule, the enterotoxins, the cytotoxic enzymes, and it causes gastroenteritis and bleeding diarrhea. The cycle starts with the bird contaminating the water using the feces, and then that water, if we use it for if we drink it straight from there, we will get infected, of course. But if that water goes to the agriculture industry or to animals, they can get infected too. I mean, those animals that will drink that water will be infected, and if, for example, it's a cow, then the milk is going to be infected too. And in the case of the agriculture, that those products that will come from that will be contaminated too. So this bacteria is very common, and as it says here, it has more cases than Salmonella and Shigella together. Treatment is radiation and mineralization, and maybe antibiotics in serious cases. And it can also create the Guillain-Barré syndrome, which is a pre-stage for the multiple sclerosis, the demyelinization of the neurons. Very, very dangerous one. And then we have the Plasmodium shigelloides. Shigelloides. Bacilli gram-negative, no spores, motile, flagella, facultative anaerobe, catalase and oxidase positive, and no lactose fermentation. It's the exception in the Enterobacteria family for the last two properties, because it doesn't fermentate the lactose and because it has oxidase positive, whereas the Enterobacteria family is oxidase negative. Because it's gastroenteritis, watery diarrhea, it requires a high infective dose, and the transmission is through raw seafood. And then we have Listeria Monocytogenes. These are bacilli gram positive, and it can aggregate in a palisade aggregation. They form like a wall, similar to a wall. And there are no spores, catalase positive and oxidase negative. It's a facultative anaerobe, and it has the umbrella mobility because of that. And then it's a psychotroph and halophil, and it creates biofilms as well. It has many virulence factors that make it resistant. It can enter the cells if it wants, and can survive the stomach acid because it has an enzyme as a protection, the glutamate decarboxylase. Low effective dose, and it can cause febrile gastroenteritis, the microbial damage, and also invasive systemic diseases, such as, because it likes to go to the brain and to the placenta, and even abortion and neonatal listeriosis. This is the invasive systemic disease is for immunosuppressed people, and the abortion and neonatal for pregnant women. We find Listeria Monocytogenes in raw dairy, fish and meat, and sprouts as well. Prophylaxis cooking at more than 75 degrees. Hygiene and be careful with cross-contamination. Then we have Aeromonas. It's a bacilli gram-negative, psychotroph, facultative anaerobe, motile with polar flagella, no halophil. It likes pH more than 6, so neutral pH, and it can create biofilms. It lives in contaminated water or food and can enter the body orally or through a wound. It has many virulence factors, but it's a high infective dose. We have cytotoxins, it can create pores in the plasmatic membrane, and cytotoxic enterotoxins, which can pull the water out of the cell. So one to create the pore and one to pull the water out, causing the gastroenteritis, nausea, vomits, bleeding, diarrhea, and even septicemia. Aeromonas, it's flesh-eating. It will eat the wound down to reach the connective muscle tissue. It will cause severe damage and pain, this bacteria. And then we have Brucella. Brucella is a Coccobacillus, gram-negative, immotile, neutral pH, strict anaerobic, catalase and oxidase positive. Only on high nutritious food is present this one. It likes milk and meat in food with high nutrients. And it's a zoonosis because it comes from the cows. Low infective dose, under 500 cells, and the transmission can be oral, by contact, air, and parenteral. Macrophages carry these bacteria as a Trojan horse to the lymph nodes, and this can generate granulosomes. Very dangerous. We find, as I said, Brucella in raw milk from sick cows or goats, because it has zoonosis. And well, to prevent the Brucella, we have the animal vaccination and as well as the milk pasteurization. And then we have viruses and prions. Virus, we have hepatitis, viral gastroenteritis, and polio. The hepatitis I, we have the P. coronavirus enteric transmission. Is very resistant, pH 1, 60 degrees, salty water, and it lives for months. It gets inside the intestine, then it goes inside the enterocytes, then to the blood, causing the viremia, then it moves to the liver, to the hepatocytes, and then internal multiplication happens, and the T cells attack the hepatocytes to kill the internal virus. So basically, it's going to kill a part of the liver to try to kill the virus inside that cell. And the bilus, they can of course enter the biliary duct. The bile sends the virus back to the gut and it will exit in feces. It causes fever, malaise, loss of appetite, and nausea. We can find it in the bivalve mollusks, contaminated water, such as in veggies too, and the prophylaxis, hygiene, cooking, freezing, and the vaccine for the hepatitis. Hepatitis E, epivirus enteric transmission, the same thing as the hepatitis A in here. It can be dangerous for pregnant women and leads to hepatic failure due to less active immune system. It's very similar to hepatitis I, and the pathogenesis and the clinical picture is the same. Then we have the viral gastroenteritis, the rotavirus, norovirus, sapovirus, enteric adenovirus, which is the only one of DNA virus. The other ones are RNA viruses, and the astrovirus, all enteric transmitted. PCR with reverse transcriptase to detect the RNA viruses is necessary for that. enterocyte invasion and destruction that will create malabsorption, and this will accumulate a lot of carbs and sodium in the lumen, and water will follow them to equal the concentrations, and that causes the diarrhea. So basically, because of the malabsorption due to the destruction of the enterocyte. Neurotransmitters are also affected by the viral gastroenteritis because the signal to the brain then will get the vagus nerve to send the signal to contract to the stomach, and that will cause vomiting. We find virus in the raw mollusks, fruits and veggies, and it's very resistant, so we need to cook it well and have an hygiene, especially hands washing. And then the polio is the only natural host of the polio virus. There is human-to-human transmission as nasopharyngeal or enteric. It grows in the nasopharyngeal mucosa and then attacks the central nervous system. It attacks indeed the spinal cord, and it causes the paralysis, which is irreversible. And it usually attacks children under 3 years of age. Fortunately for us, we have an effective vaccine against polio. Prions are misfolded proteins, and they have incubation periods between 20 and 30 years. That's a lot of time. They create damage to the central nervous system, developing the subacute spongiform encephalopathy that can be fatal. Transmission is through contaminated food, heredity, or by a spontaneous mutation. The mad cow disease is caused by a prion, and prions are rich in beta sheets. They are insoluble and they aggregate themselves, if you remember from the first video. Intestine to the payer plate, to the lymphatic system, to the brain. And once in the brain, they are going to bind to normal proteins to convert them into prions. And then once we have a considerable amount, they will aggregate, creating fibrils, and fibrils will inactivate neurons. We will feel a gradually loss of muscle control, coordination, and an increased depression and insomnia. Estimated death after a year. Prions can be found in meat products with a lot of nerves, such as the spinal cord or the brain of the nerve, the eyes as well, of infected animals. Prevention, control on the farm, in the slaughterhouse, and in the meat industry. They can resist to formaldehyde and UV light and to 80 degrees as well. So we need to to kill them, use the autoclave at 134 degrees Celsius. Okay, so if we move on, we have the microbiological monitoring of foodstuff. We have to follow a microbiological criteria, and we have two types of methods here. The safety ones for bacteria and toxins, and the quality ones. The quality ones are that are meant, and the quality ones for the hygiene of the processes. They determine if the processes of the creation of the product have been done correctly. In fact, this applies to the product at the factory, and this is in the safety one is in a product we find in the supermarket already in the market. If it fails this criteria, we have to remove from the market the product because it's not edible and not safe. If we fail the quality criteria, the hygiene of the processes, we have to check the production processes and improve them because it's still in the factory. So the contents of the microbiological criteria are: we need to say the found microorganism, the method we use, the number of samples, the microbiological limit on that food we analyzed, the analyzed food of course, the number of analytical units, and if we analyze a product that's already in the market or at the factory, and their respectively actions if it doesn't comply with the criteria. For safety, we already discussed, remove it, and for hygiene of the processes, well, to improve them. How to take samples? It has to be a representative of the whole lot and identifiable. Has to be protected from contamination and stored in an appropriate temperature. The analysis has to be within 36 hours. If it passes the 36, we have to take another sample. And for finished products, we just use the original sealed container, or if it's in bulk, we need to put them in a sterile container. And in the production line, the finished product is for the safety one, and the production line is for the quality, hygiene of the processes. In this example, we need to get a little bit all over the place, if not from a phase only. We have to store the non-perishable ones at ambient temperature, the refrigerated, at 0.4 degrees, and the frozen at -20 degrees Celsius to preserve them as much as possible before the analysis. Then we have the conventional methods of microbial identification. We have a bunch of methods, but we're going to view the conventional or classical methods. So first, we need to obtain the pure culture of the microorganism. And here we have to differentiate if you want to count or to detect the microorganism. So if we want to count, we can do a direct isolation. But if we want to detect it in the food, we have to do an enrichment, which is a primary enrichment, the reactivation of all the microorganisms present in that sample, and as a secondary enrichment for the selection. This has to be a selective one. So the first one is to let all the bacteria, all the microorganisms grow in that sample. And the second one is to select the microorganism we want to detect. So we put it on a selective culture medium so it can grow alone and is going to be easily to identify, to detect. Then we have to identify the colonies. Then we have to observe the morphology of them using the gram stain. And then we have to do the identification tests. We have the primary tests that detect the taxonomic gender, and the secondary ones that detect the species. The primary ones are the catalase with H2O2, and if there are bubbles, then the catalase is positive, and if there are no bubbles, then it's negative. Then we have the oxidase. We have to use a reactive, and if it's oxidized, then it's purple, and if it's not purple, then it's not oxidized, and it's oxidase negative. The reduced is clear. And the coagulase as well, we use a plasma sample, and if it coagulates and with white dots, well, it's a coagulase positive. If you remember from the previous video and the beginning of this one, the catalase positive, oxidase negative were the classification of the various families we discussed of the pathogenic bacteria mainly. And the classification comes from these tests. They are very fast because they only need short time. Indeed, this is only adding H2O2 and see if there are bubbles or not. This is very, very fast, as well as the others. Then we have the secondary ones and tertiary as well, such as lactose fermentation, the methyl red and Voges proscare, the decarboxylation of the amino acids, and the citrate as a carbon source. And they're very, very long time for them, even more than 24 hours. So to not do one at a time, we use an API multi-test system. And with this system, we can do a lot of these tests at the same time. So in 24 hours, we will have the result for more than one of these tests. Otherwise, it will take like 5 days to complete them all. Okay, that's it for this video. The hygiene and sanity of food is done. I hope you understood well all of this. It was a huge video. I had to split it in 2 parts. And this diagram is going to be down below in the description. It's on my website. You can download it for free. And like the video down below, subscribe to the channel if you're new, and click the bell icon below so you will be notified every time I upload a new video. Thank you so much for watching, and see you on the next one. Bye!